Let's start with the chapter 10, the cell cycle and cell division. So let's proceed now further. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells. Indeed, of all living organisms, all cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell and eventually divide into two daughter cells is termed as cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. DNA synthesis occurs only during the one specific stage in the cell cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Next is phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycles can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through a cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases, interphase, M phase, or mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is significant to note that in the 24 hours average duration of a cell cycle of a human cell, cell division properly lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosome, karyokinesis, and usually ends with division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase, through called the resting phase, कौन सा phase? Resting phase कौन सा कहलाता है? Interphase is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three phases. G1 phase, that is gap 1, S phase synthesis, G2 phase gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. Interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continues grows but does not replicate its DNA. As or synthesis phase marks the period during which DNA synthesis or replication takes place. During this type, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, that it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid or 2n number of chromosome at G1, even after S phase, the number of chromosome remains the same, that is 2n. Now, there is a box in yellow color. How do plants and animals continues to grow all their lives? Do all cells in a plant divide all the time? Do you think all cells continues to divide in all plants and animals? Can you tell the name and the location of tissues having cells that divide all their life in higher plant? Tell. Socho. Do animals have similar meristematic tissues? Can you tell the name and the location of tissues having cells that divide all their life in higher plants that are meristematic tissues? Okay. So the next is, in animal cells, during the S phase, DNA replication begins in the nucleus and centriole duplicates in the cytoplasm. कहाँ पे डुप्लीकेट होता है सेंट्रियोल? साइटोप्लाज्म में. अंडरस्टूड? स्पीड में तो नहीं चल रहा है ना ज़्यादा, एक स्लो कर लेना स्पीड अगर स्पीड में ज़्यादा लग रहा हो तो. ड्यूरिंग द G2 phase, protein are synthesized in preparation for mitosis while cell growth continues. During G2 phase, protein are synthesized in preparation for mitosis while cell growth continues. Some cells in the adult animal do not appear to exhibit division, example heart cells, and many other cells divide only occasionally as needed to replace the cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase to enter an inactive stage called quiescent stage, G0, of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. Is only seen in the diploid somatic cell, mitotic cell division. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybees. Against this, the plants can show mitotic division in both haploid and diploid cells. From your recollection of examples of alternation of generation in plants in chapter 3rd. Identify the plant species and stage at which mitosis is seen in haploid cells in the chapter 3. So, you have studied mitosis in onion root tip cells. It has 16 chromosomes in each cell. 16 chromosomes in each cell. Can you tell how many chromosomes will the cell have at G1 phase, after S phase, and after M phase? Also, what will be the DNA content of the cell at G1, after S, and at G2 if the content after M phase is 2C? चलो बताओ, आ गया तगड़ा क्वेश्चन, आ गया ना तगड़ा क्वेश्चन. अब करो आंसर बच्चों, आंसर करना पड़ेगा ना. क्वेश्चन पढ़ते हैं वापस से. You have studied mitosis in onion root tip cells. It has 16 chromosomes in each cell. Can you tell how many chromosomes will the cell have at G1 phase, after S phase, and after M phase? Means the cell G1 phase में कितने होंगे? After S phase में कितने होंगे? Tell me the answer. So the solution is, it is given that the number of chromosomes present in each onion root tip cell is 16. As the number of chromosomes in the cell will be 16 at G1 phase, after S and after M phase. Also, if the DNA contained after M phase is 2C, hence the DNA contained at G1 phase will be 2C. It will be 4C after S and G2. Okay, understood? The number of chromosomes in the cell will be 16 at G1 phase, after S phase and after M phase. It will be 16 only. So now let's proceed further. M phase. This is the most dramatic phase of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cell is the same, it is also called as equational division. Though, for convenience, mitosis has been divided into 4 stages of nuclear division, karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves prophase, metaphase, anaphase, telophase. PM80. First is prophase. Prophase, which is the first stage of karyokinesis of mitosis, follows S and G2 phase of interphase. In the S and G2 phase, the new DNA molecule formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. Means अलग होना, ठीक है? The chromosomal material become untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during S phase of interphase, now begin to move towards opposite poles of the cell. The completion of the prophase can thus be marked by the following characteristics events. Chromosomal. The completion of prophase can thus be marked by the following characteristics event. Chromosomal material condenses to form compact mitotic chromosome. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. Centrosome, which had undergone duplication during interphase, began to move towards opposite poles of the cell. Each centrosome radiates out microtubule called esters. The 2 esters together with spindle fiber forms mitotic apparatus. The 2 esters together with spindle fibers forms mitotic apparatus. कौन फॉर्म करता है mitotic apparatus? 2 esters together with spindle fibers. Understood? Cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear envelope. So the next phase is metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of the mitosis. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis, that is metaphase, as the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosome is most easily studied. ये तो बहुत पूछा हुआ क्वेश्चन है, हर मॉक टेस्ट में कहीं ना कहीं मिल जाएगा. Best stage at which morphology of chromosome is most easily studied is metaphase. At this stage, metaphase chromosome is made up of 2 sister chromatids which are held together by the centromere. A small disc-shaped structure at the surface of the centromeres are called kinetochores. This structure serves as the site of attachment of spindle fibers formed by the spindle fibers to the chromosome that are moved into position at the center of the cell. metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. I am repeating it again. Hear it properly, okay? ध्यान से सुनना. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. Means दोनों pole से खींच जाओ. The plane of alignment of the chromosomes at metaphase is referred to as metaphase plate. The key feature of the metaphase are spindle fiber attached to kinetochores of chromosome. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. So the next is anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously and the two daughter chromatids, now refers to as daughter chromosome of the future daughter nuclei, begin their migration towards the opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole, and hence at the leading edge with the arms of the chromosome trailing behind. And hence at the leading edge, the arms of the chromosome trailing behind. Means trailing behind means पीछे पीछे. Thus, anaphase stage is characterized by the following key events: centromeres split and chromatid separate. Centromere split and chromatid separate. Chromatid move to opposite poles. Next phase is telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosome can no longer be seen, and each set of chromatin material tend to collect at each of the two poles. This is the stage which shows the following key events: chromosome cluster at opposite spindle poles, and their identity is lost as discrete element. Their identity is lost as discrete element is the key event of telophase. Nuclear envelope develops around the chromosome cluster at each pole, forming the two daughter nuclei. Nuclear envelope develops around the chromosome clusters at each pole, forming two daughter nuclei. Nucleolus, Golgi complex and endoplasmic reticulum are reformed. Next is cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. Mitosis accomplishes not only the segregation of duplicated chromosome into daughter nuclei, that is karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. फूरो बनता जाएगा और साइटोप्लाज्म को डिवाइड कर देगा टू में. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation starts in the center of the cell. In plant cells, wall formation starts in the center of the cell, and grows outward to meet the existing lateral wall. कहाँ पे ग्रो करेगा? आउटवर्ड में. चेंज करके इन्वर्ट कर देंगे बट करेक्ट है आउटवर्ड. अंडरस्टूड? टू मीट द एक्सिस्टिंग लैटरल वॉल. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that represent the middle lamella between the walls of two adjacent cells. जो सेल प्लेट होती है रिप्रेजेंट करती है मिडिल लैमेला between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organelles, karyokinesis is not followed by the cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytin. Example, liquid endosperm in coconut. So let's proceed further. Next is significance of mitosis. Mitosis or the equational division is usually restricted to the diploid cells only. Important line है ये, only आ गया इसलिए. Mitosis or the equational division is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. However, in some lower plants and in some social insect, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about haploid and diploid insect? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. Mitosis usually results in the production of diploid daughter cells with identical genetic complement. यहाँ पर चेंज करके नॉन आइडेंटिकल भी कर सकता है बट करेक्ट है ओनली आइडेंटिकल. मीन्स आइडेंटिकल आएगा करेक्ट में विद आइडेंटिकल जेनेटिक कॉम्प्लीमेंट. द ग्रोथ ऑफ मल्टीसेल्युलर ऑर्गेनिज्म इज ड्यू टू माइटोसिस. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and blood cells. The cells of the upper layer of epidermis, cells of the lining of the gut, and the blood cells are being constantly replaced. Mitotic division in the meristematic tissues, the apical and the lateral cambium, result in the continuous growth of plants throughout their life. Mitotic division in the meristematic tissues, the apical and the lateral cambium, result in a continuous growth of plants throughout their life. Meiosis, the production of offspring by sexual reproduction, includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phases in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key feature of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis first and meiosis second, but only a single cycle of DNA replication. Meiosis first is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis first is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosome and recombination between non-sister chromatids of homologous chromosome. Recombination between non-sister chromatids of homologous chromosomes. 4 haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped under the following phases: meiosis first, K phase, prophase first, metaphase first, anaphase first, telophase first. Meiosis second, K phase, prophase second, metaphase second, anaphase second, telophase second. So now we are going to read meiosis first का पहला फेज प्रोफेज फर्स्ट। यहाँ से भर भर के क्वेश्चन आते हैं, कोई भी ईयर हो, कंफर्म श्योर शॉट। सो लेट्स स्टार्ट दिस विथ फुल कंसंट्रेशन। Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior, that is lactotine, zygotine, pachytine, diplotin and diakinesis. During lactotine stage, the chromosome become gradually visible under the light microscope. यहाँ से क्वेश्चन आया था 2024 में और वो मेरी आत्मकथा है, बस पूछो मत। Again I am repeating, during lactotine stage, the chromosome become gradually visible under the light microscope. The compaction of chromosomes continues throughout lactotine. This is followed by the second stage of the prophase first called zygotine. During this stage, chromosomes start pairing together and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrograph of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptonymal complex. The complex formed by a pair of synapsed homologous chromosomes is called a bivalent or a tetrad. However, these are more clearly visible at the next stage. The first 2 stages of the prophase first are relatively short-lived compared to the next stage, that is pachytine. During this stage, the 4 chromatids of each bivalent chromosome become distinct and clearly appears as tetrads. This stage is characterized by the presence of recombination nodules, the site at which crossing over occurs between non-sister chromatids of the homologous chromosome. This stage is characterized by the appearance of recombination nodules, the site at which crossing over occurs between non-sister chromatids of the homologous chromosome. किसमें होती है? Non-sister chromatids of the homologous chromosome. Crossing over is the exchange of genetic material between 2 homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called recombinase. Crossing over leads to recombination of genetic material on the 2 chromosomes. Crossing over leads to recombination of genetic material on the 2 chromosomes. Recombination between homologous chromosome is completed by the end of pachytine, leaving the chromosome linked at the sites of crossing over. Recombination between homologous chromosomes is completed by the end of pachytine, leaving the chromosome linked at the site of crossing over. The beginning of diplotin is recognized by the dissolution of the synaptonymal complex and the tendency of the recombined homologous chromosome of the bivalents to separate from each other except at the site of crossovers. The beginning of diplotin is recognized by the dissolution of the synaptonymal complex and the tendency of the recombined homologous chromosome of the bivalent to separate from each other except at the site of crossovers. These X-shaped structures are called chiasmata. In oocyte of some vertebrates, diplotin can last for months or years. महीनों से लेकर सालों तक. The final stage of meiotic process first is a diakinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents transition to the metaphase. Next is metaphase first. The bivalent chromosome align on the equatorial plate. The microtubule from the opposite poles of the spindle attached to the kinetochore of homologous chromosome. Next is anaphase first. The homologous chromosome separate while sister chromatids remain associated at their centromere. Telophase first. The nuclear membrane and nucleolus reappear. Cytokinesis follows and this is called as dyad of cells. The nuclear membrane and nucleolus reappear. Cytokinesis follows and this is called as dyad of cells. Although in many cases the chromosome do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic divisions is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. It's correct, true statement. There is no replication of DNA during interkinesis. Statement 1 true, statement 2 false में आ सकता है, ठीक है. यहाँ पे there is replication of DNA during interkinesis आ सकता है, तो ये false है. True है, there is no replication of DNA during interkinesis. This series will help you a lot. Interkinesis is followed by prophase second, a much simple prophase, then prophase first. Next is meiosis second. Prophase second. Meiosis second is initiated immediately after cytokinesis, usually before the chromosome have fully elongated. In contrast to meiosis first, meiosis second resembles a normal mitosis. The nuclear membrane disappears by the end of prophase second. The chromosome again becomes compact. Metaphase second. At this stage, the chromosome align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochore of sister chromatids. Next is anaphase second. It begins with the simultaneous splitting of centromere of each chromosome, which was holding the sister chromatids together, allowing them to move towards opposite poles of the cell by shortening of microtubules attached to kinetochores. Next is telophase second. Meiosis ends with the telophase second, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells. Meiosis ends with telophase second, in which the two group of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells. Next is significance of meiosis. Meiosis is the mechanism by which conservation of a specific chromosome number of each species is achieved across generation in sexually reproducing organisms. Even though the process per se paradoxically results in reduction of chromosome number by half, it also increases the genetic variability in the population of organisms from one generation to the next. Variation are very important for the process of evolution. Variation are very important for the process of evolution. Understood? Next is summary. Nec speedy way. According to the cell theory, cell arises from pre-existing cells. The process by which this occurs is called cell division. Any sexually reproducing organism start its life cycle from a single-celled zygote. Cell division does not stop with the formation of the mature organism but continues throughout its life cycle. The stage through which a cell passes from one division to the next is called cell cycle. Cell cycle is divided into two phases called interphase, a period of preparation for cell division, and mitosis, M-phase, the actual period of cell division. Actual period of cell division होता है mitosis, M-phase. Interphase is further subdivided in G1 S and G2. G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occur during this phase. In G1, S phase marks the phase of DNA replication and chromosome duplication. G2 phase is the period of cytoplasmic growth. Mitosis is also divided into 4 stages, namely prophase, metaphase, anaphase, and telophase. One second, कुछ याद आया है तो वो लाइन आपको मैं वापस से सुना देता हूँ. मेरे को एक लाइन याद आई है, ठीक है? वेट ए मिनट. कौन सी लाइन थी वो? प्रोटीन सिंथेसिस वाली, ठीक है? सो क्वेश्चन था, क्या था क्वेश्चन? बता रहा हूँ. 1 मिनट. With the cell diploid, during the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. प्रोटीन का सिंथेसिस होता है G2 में, ठीक है? अंडरस्टूड? प्रोटीन का सिंथेसिस होता है G2 में. So now let's proceed and continue with that only. Mitosis is also divided into 4 stages, namely prophase, metaphase, anaphase, and telophase. Chromosome condension occurs during prophase. Simultaneously, the centrioles move to the opposite poles. The nuclear envelope and the nucleolus disappear, and the spindle fiber start appearing. Metaphase is marked by the alignment of the chromosome at the equatorial plate. During anaphase, the centromere divide and the chromatid start moving towards the 2 opposite poles. Once the chromatid reaches the 2 poles, the chromosome elongation starts. Nucleolus and the nuclear membrane reappear. This stage is called telophase. Nuclear division is then followed by the cytoplasmic division and is called cytokinesis. Mitosis thus is the equational division in which the chromosome number of the parent is conserved in the daughter cells. In contrast to mitosis, meiosis occurs in the diploid cells. Which are destined to form gametes. It is called the reduction division since it reduces the chromosome number by half while making the gametes. In sexual reproduction, when the 2 gametes fuse, the chromosome number is restored to the value in the parent. meiosis is divided into two phases, meiosis first and meiosis second. The first meiotic division, the homologous chromosome pair to form bivalents and undergo crossing over. Meiosis 1 is long prophase, which is divided further into 5 phases. These are lactotin, zygotin, packaging, diplotin, and dikinesis. During metaphase first, the bivalent arranged on the equatorial plate. This is followed by anaphase first, in which homologous chromosome move to the opposite poles with both their chromatids. Each pole receives half the chromosome number of the parent cell. It's true statement. In telophase first, the nuclear membrane and nucleolus reappear. Meiosis second is similar to mitosis. During anaphase second, the sister chromatids separate. Thus, at the end of meiosis, 4 haploid cells are formed. With this, we completed the chapter. Thank you for hearing.